Well, hello. This is not going to be a typical hangout that I usually do. This is just going to be a hangout that I'm going to get some people in here, hopefully, that might know a little bit about biochemistry and biology to help me with some presentation that we're going to be working on. We're doing a series, DNA to protein, basically an anabolic way of doing it from the ground up so people can understand when people are talking about this stuff, what actually they're talking about. What is a nitrogenous base? What is a nucleoside? What is a nucleotide? People have been at, oh shit, hang on. I'm getting feedback here. One sec.
Better?
Much better. Yeah, that's about the third time I've done that so far. You would think by now after doing these live streams, I remember to turn off the other video, but I have that video up so I can actually watch the live chat. Not that this is going to be of any interest to anybody, because like I said, this is just a hangout to chill and talk about biochemistry a little bit, and kind of give me some ideas to add to my presentation. I've already literally kind of finished the part one as far as the slides, but I wanted to get some feedback on it. As far as the technical information on it, and just what in general I can change it up. So because there's a fine line between it being informative and being too simplistic and being too complicated. Because the whole point is, when I watch a lot of videos on these subjects, I kind of find that most of these videos are either A, really too technical, or B, too simplistic. And there's nothing in the really middle ground that says, you know what are these words that they're talking about? What does all this mean? And I wanted to kind of demystify it for people. And even though this is not my area, it's something I'm decent at, so maybe I figured I'd give it a shot. So especially if you know about biochemistry or genetics, come on in and help me out with this. Even if not, come on in anyways, shoot with me. I'm kind of bored. So do you kind of want to see what I got so far? Like I said, I don't want to like get into it if there's not many people to help me with it. So I'll just chill out till people come in, but I'll show you. I'll just kind of show you a little bit what I'm doing as an overview there, of course. Okay.
Okay.
Okay. Can you see it?
No. Oh, yeah, now. DNA
Yeah.
and proteins, too.
Yeah. So this is kind of my presentation so far. I'm keeping it pretty simple. I did pretty much 99% of this all on my tablet. I didn't even do it on a computer, so it's a little bit rudimentary, but I think it might get the point across.
Aye.
But what I was going to do is I was just going to go through the slides here, and then as people join us, kind of get ideas of what I can change on it.
Hey, when you're talking about proteins, are you talking about life starting with the protein or something like that? Or
No, proteins are different things consisting of amino acids that have different things they do, like enzymatic functions,
Aye.
catalyzation, structural proteins like collagen.
Aye.
So what the proteins will be produced from is from DNA. It's from your actually coding your genes. So
Aye.
what I want to do is I want to do a 3-part, maybe 4-part series, starting with the DNA, then going to mRNA, then going to transport, then going to proteins, which is basically transcription, mRNA, and translation is called. And then maybe do one for DNA replication, because that's actually still part of the central dogma. But that requires a different video that deals with different DNA polymerizations and things like that. That probably would be a little more technical than I ever need to get into, because it's
I'm probably not going to understand what you're talking about. So.
Well, that's kind of good, because I've actually need somebody here that doesn't know this stuff. So when we're explaining to them, if I can explain to somebody who has absolutely no clue, then I think that then the presentation is a success. Obviously, I mean, this is the whole point of demystifying these crazy terms. I mean, you hear people using terms like ATP synthase, right? Well, what the hell is ATP synthase? What's a synthase, or a transferase, or a kinase, or a phosphorylation? What is that? And so these words people get confused on what they actually mean. So I was going to kind of give an example, give a breakdown of how to remember what these words mean and take the sting out of them, because they sound so technical. And then as I get more, further down the series, we'll get more into it and demystify some of the bigger words. Like, if I say like aminoactyl tRNA synthase, people are like, well, what the hell is that? But it's really not that complicated. With the basically, I guess you could say.
Aye.
Anyways, I guess I'll kind of go over this a little bit since I've got you here. Like I said, I don't expect too many viewers on this because it is not that kind of video, but I thought I'd make it live anyway. So let me know if anybody comes in. So basically, I'll just start the start stuff. Okay, so I thought this came out pretty good.
What are you talking about?
Oh, you can't see it. Yeah, good job, Steve. Yeah, one sec.
Oh, you're talking about your PowerPoint presentation,
Yeah,
right?
yeah. Let me screen share one sec.
I
Okay, now you can see it, right?
Yes.
Okay, I thought it came up pretty good. It's supposed to have this curtains come up, but it doesn't show on this computer. I don't know why. I have to figure that out.
Demystifying basic biochemistry and protein synthesis.
Yeah. So what I wanted to do, I wanted to start out just basically what is DNA, and
Uh,
and this is for somebody who has no idea what any of this stuff is right here. I mean, this
uh,
is
it's the building block, right?
right, but I mean, do you know what? I mean, do you know what these are and these are and this is and this?
Well, I don't know what are you referring to?
Can you see it?
Uh, no.
You can't see the screen.
Yes, I can, but
Oh,
I don't know what you're referring to.
you don't see my mouse?
Well, it's very small, so
Oh, that's why. Um, well, basically, it's going to be the ribose, the phosphates, the nitrogenous bases, the hydrogen bonds, that kind of stuff.
I,
So I basically just, I'm explaining what DNA stands for, deoxyribonucleic acid, which I'm actually I need to add something here. I'm going to put in basically explain why it's an acid, and which we'll actually go into later, why it's an acid.
okay.
A lot of people ask, why is DNA an acid? Well, an acid is anything that donates a hydrogen ion, a proton, basically. Same thing. And
Yeah.
that's why it's called an acid. But DNA is a polymer comprised of nucleosides, which are nitrogenous bases with a 2-deoxyribose sugar. Again, sounds complicated. And what I'm going to do is I'm going to explain to them what a polymer is, which I still need to add. I forgot to add that. So I'm going to explain what a polymer is from a monomer. And then I'm going to break down and explain what nucleosides and nitrogenous bases are, what actually a 2-deoxyribose sugar is, explain nucleotides or nucleosides with a phosphate group, and then I just threw this one in there that two types of DNA exist in the human body, nuclear DNA and empty DNA, which is mitochondrial, because we're going to be talking about nuclear DNA. Okay? So that's just a basic overview of what we're going to be doing. Got that?
Yeah, you kind of confused me with nuclear DNA. Is it like atomic?
No, nuclear DNA is found in the nucleus of the atom. The nucleus of the cell. Sorry, nucleus of the cell. So there's in your cell, you have what's called the mitochondria. The mitochondria.
The power station or what?
Yes, the powerhouse. Yes, for metabolic respiration. And that has empty DNA in it, which is mitochondrial DNA. We're not going to be talking about that. We're going to be talking about nuclear DNA, which is the DNA that's found in every nucleus in your entire body. Regular DNA, for the most part.
Which is like the center of the cell, right?
Yes.
Okay.
Okay. So this is what the central dogma of biology is. Basically, it's replication, right? And you know that a gene goes through mitosis. Obviously, a cell goes through mitosis.
Yes.
The DNA replicates.
Yeah.
So this will be part 4 in my series. This is going to be 1. This is going to be 2. This is going to be 3 over here.
Actually, I get confused with like mitosis and meiosis.
The easiest way to explain that, the way I always remember, what process created me? Well, that was meiosis. A cell that takes, divides and creates a haploid.
Yeah.
Okay, like a sperm or an ovum, it creates a haploid gamete. That's how I remember that. So that meiosis is sexual reproduction like that, and then mitosis is your cells just splitting and then replicating themselves.
Okay.
Yeah, that's an easy way to remember that.
All right.
There's various phases, which if we want to do a series on cell division later, we could. I would have to review that. I haven't reviewed that in ages, but you have the metaphase, prophase, interphase, anaphase. Remember that stuff?
Not really.
Yeah. There's a lot to it because of the spindles and it's complicated. But anyways, this is going to be about DNA. So I'll do the replication afterwards, and then transcription to DNA to mRNA, post-processing mRNA, transport of mRNA to the ribosome, and translation of the mRNA to amino acid chains of protein. So this is actually what we're going to be going over. This kind of little heads up. This video here actually doesn't deal with any of these. This video here is just going to be on the creation of DNA. Okay? And like I said, this is not actually how it's built in the body, but this is a more organic approach to it. But it's close. Anybody else come in here yet?
Uh,
No?
I don't know.
Hang on, 1 sec. I got the cameraman on anyways. 1 sec.
Usually you'd have someone else also, but
Let me put cameraman on just in case. I don't want to get bombed. Guess it's going around now. Okay. Can you still see?
yeah,
Okay.
nitrogenous
Nitrogenous.
bases.
Yeah, some people say nitrogenous. I like the word nitrogenous. But
Okay.
I'm American, so we have funny
Like
words.
androgynous, I guess.
Yeah,
Alright.
exactly. And what we're going to do is we're going to be explaining basically this is the foundation. Everybody kind of remembers A, T, G, and C, right? Those
Yeah,
are
for DNA.
Yeah, for DNA. So what are A, T, G, and C? They are called nitrogenous bases.
Okay.
They're produced by I'm not going to go into the metabolic pathways of how they're produced, but because that's complicated and we have a whole different video. But basically, I just wanted to show people how they're produced. They are produced metabolically. Deamination, which is the removal of an amine group, such as histone or uracil. Again, not going to get into this video that much, but I do plan to explain that later on.
Aye.
And actually, I kind of do it basically here. It's just here's a breakdown of what these things look like. Okay, this is cytosine, uracil, and thymine, which are called pyrimidines. Okay,
Aye.
guanine and adenine, which are called purines. And basically, you can see there's a structural difference. These are called double bonds. These are, and every one of these little nodes here, these little apexes, have a carbon atom right there. So these are called a carbon ring.
Aye.
So when it says deamination, the removal of the anamine group, this is your amine group right here, the NH2. So it just removes that and puts an oxygen double bond there. And so cytosine will actually convert to uracil, but it is a little bit of a process, which I'm not going to get into in this video, but I just want people to know that. So what I want to get into is DNA generally consists of A, T, G, and C, while RNA consists of A, G, C, and U. So RNA, which is ribonucleic acid, has uracil instead of thymine. That's the main difference. And I say generally because there are other things that can be in DNA and RNA, but we're not going to get into those. I decided not to. I was going to talk a little bit about them, but then I realized, yeah, you don't really need to know about them just to make DNA. So,
all right.
The way they pair up is a pyrimidine always will base, should always base with a purine here, right? So thymine will pair with adenine, and then cytosine will pair with guanine, okay?
Aye.
Okay, so these are what the basic things, these are the basic building blocks we're going to be using to build the DNA. Cytosine, thymine, adenine, and guanine. Uracil, again, is only for RNA. So we're going to build it up. And the way we're going to build it up is basically by demystifying the terminology first, okay?
Aye.
Going over the prefixes, we have mono, single, di, double, tri, triple. Poly means many. Synth is synthesis. Ase is an enzyme. D is a pyrimidine. Ine is a purine. Okay?
Aye.
And I'm doing this really fast because this is not my presentation. I just want to explain quickly. But when I actually do my presentation, it'll be more polished, obviously. But okay, so these are your bases. Remember those bases we start off with? A, C, A, G, and T?
Yeah, the nitrogenous.
Nitrogenous?
Sorry.
Okay, so when we have a nucleoside, we're going to be creating a nucleoside, which is these, and then we're going to be explaining what that is to how it relates to a nucleotide. But basically, if you start off with the base, like cytosine, when you make a nucleoside, it'll be called cytidine. Okay?
Aye.
And so if it has D-I-N-E, you remember it's a pyrimidine, right? And if it's I-N-E, it's a purine. So these are my two purines, A and G, and then my two pyrimidines, G, C, and T.
Aye.
So basically the nomenclature goes, if it's mono, it has one phosphate. If it's di, it has two phosphates. And if it's tri, it has how many?
Three.
Three phosphates. Actually, I made a mistake here. See, this is why I'm going ahead. See, I need to change this to D right here. So let me fix it. Mark, this is why I'm going over it because I need to fix these minor mistakes. This should be cytidine.
So you're just telling me how the DNA is put together?
Yeah, I'm going to be building up the DNA here.
Okay.
But this is the nomenclature. Okay, so it starts off with the base, and then it goes to the nucleoside. Okay.
Okay.
And if it's a nucleoside, will always end in D-I-N-E or I-N-E. And then how many phosphates it has. That's how we break these down. So guanine will be guanoside, And then if it has one phosphate, it would be monophosphate. 2 would be diphosphate. 3 would be triphosphates. Okay. And then I wanted to get into A's a little bit because these sound like technical words.
Okay.
But they're really not. So if A's is an enzyme, right, and a protein does an enzymatic function. Poly means many. What do you think polymerase means?
Poly what?
Polymerase.
Many enzymes.
Yeah. Well, basically, it's actually in this case it's a polymeric chain. It's a polymer of enzymes, and the polymerase is what's used to actually do the translation.
Like plastic?
No. Well, no, kind of. Plastic is a polymer as well. That just means it has a bunch of things stuck together rather than being individual monomers.
Okay.
And then synthase, another people hear all the time, like ATP synthase. So synthesis, it usually is a catalytic function of enzymes. So when you hear synthase, you know you're dealing with an enzyme, and you know you're dealing with synthesis. So synthase is an enzyme that helps in synthesis. It helps, is a catalyst. Okay. I gotta add kin here, by the way. See, this is why I'm doing this, because I need to utilize my boo-boos. Kinase. Kin means actually emotion. Kinetic energy, motion, right? So kinase, what do you think that is? If it's motion and an enzyme, what does it imply to you?
You just said what it was.
Yeah, it does. It's an enzyme that does motion. It moves something around.
Okay.
So what do you think transferase is?
It transfers one to the other in enzymes, or what?
Yes, it's an enzyme that transfers something. Transferase. See, this is why it gets easier. Okay, how about if I say phosphotransferase?
It transfers phosphor.
Yes.
Enzyme?
Transfers phosphor. So it's a transferase. It's an enzyme that transfers phosphor, which is phosphor.
I get it.
So that's how you read these things. In phosphatase, if we add a phosphate to something and we want to remove something, like the phosphate, if we want to move it, we call it phosphatase. Okay, so phosphorotransferase adds a phosphorus group. Okay, called phosphorylation, and then if we want to remove it, we have phosphatase.
Aye.
So this is how we break down words. Okay.
Aye.
The next one I'm going to do is explaining the sugar, because there's two types of sugars. Obviously, there's going to be the one used in DNA, and there's going to be one used in RNA. We're going to be focusing on the one on DNA.
Okay.
But you got to know the numbering of it. And the way this is numbered is, this is a carbon. These are all carbons at the apex. So the carbon starts at the first position from the O, which is oxygen. So we have 1, 2, 3, and 4. Okay? That's a very simple way. 1, 2, 3, and 4. So the position we care about right now is the 2'. And sugars use the prime number. So if you hear 5', you know you're dealing with a sugar. If you just see the number 5, that just means it's a carbon.
You're talking about prime numbers as like only divisible by itself in 1?
No. Prime is just when you say 5', it has that little prime mark. It means it just means we are working with a sugar.
Aye.
Okay. And the 2' here is where it's going to be the difference between a deoxyribose and a regular ribose. Well, a pentose ribose, I should say. So this right here is going to be the difference, and I'll explain that as we go on. Okay.
Aye.
So if I say the 5th carbon in the sugar, the 5', we're talking about this 5 up here.
Aye.
Okay. So now we're going to start building this stuff up here.
Aye.
One of the things we need to talk about, I'm going to get into, which I may actually drop this down and throw it in later, but the hydrogen bonds. People ask, well, why does guanine and cytosine go together, and why do adenine and thymine go together? Well, you can see the way the structure is. Even if you don't even know anything about what any of this means, you can see that there's 3 nodes here, right? And there's a hydrogen bond that forms between the double pair bond oxygen here, nitrogen and nitrogen. And here's the same thing. So that's why these go together. They form what's called hydrogen bonds. Okay. And I need to add another video to really explain more hydrogen bonds, which I'll do later. But just for personal reference now, this is why they go together, because G to C have 3 hydrogen bonds, and A to T have 2 hydrogen bonds. Okay.
And A goes to G and T goes to C.
A goes to T, G goes to C.
Oh, I got that mixed up. Okay.
Yeah. And it backwards. And then this is another version of it, so you can see that there's different ways of doing these diagrams, okay?
Aye.
All right, so now we're going to actually build them up. So the first thing we have is that we have a, actually, you know what, let me go back. I need to do something real quick. This is why I'm doing this as we speak.
Aye.
One sec. That's that. Yeah. All right, let me do this for you. Is anybody else join us yet? No. Nobody loves me. It's okay.
It might be because of me. I guess people don't. Maybe they know I'm here. I don't know.
Whatever. All right, so this is how we're going to build this up. This is how we start building DNA. What we have here is adenine, okay, and we have a sugar. So when we have what's called a nucleoside, as we said at the beginning, is going to be a nitrogenous base with a sugar group.
Aye.
So this is my purine, my nitrogenous base, and this is my sugar. Okay. Now we're going to put them together, and they use big words which we're going to demystify here.
Aye.
They call this a beta N9 glycosidic bond.
Okay.
Okay. Doesn't really be as complicated as it sounds. Okay. Beta just means that this group that's going to be here normally is on the same side as this. They're on the same side of the sugar. If it's alpha, it's going to be on the opposite side. So this H is going to be over here rather than down here. Okay. This is normally going to be an OH right here. If you look, so I gotta show the previous picture for sugar. See, I have HOCH2.
Mm-hmm.
And I'll modify this so I can do this during the presentation. And see, I have the OH here.
Okay.
These are on the same side. They're on the top part of it, right?
And the A is on the other, right?
Yeah, the H is on the other. So if these two on the same side is considered a B bond or a beta bond, if this is OH group, they're called hydroxyl group, is over here, it's an alpha bond. Okay, that's it. That's why they call it B. Now, why they call it an N9 is that it is a nitrogen atom here, and it's at the 9th position of the carbon. Remember about the numbering? We're going to number this purine starting from over here. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this 9th position is going to be on the first position of the ribose. So we call that a nitrogen 9 glycosidic bond.
Okay.
Okay. If it's on a pyrimidine, it's going to have the 1 position here, and it's going to be a 1-1 bond, so you just leave that off. So it's just considered a B or beta glycosidic bond. Okay, so it's linked by a beta 1,9 nitrogen glycosidic bond for purine, or a B9 glycosidic bond for it's 1 carbon by base to the carbon to sugar for pyrimidine linkage. So this is your base, your nitrogenous base, and this is your sugar. This is a nucleoside. And how these come together is pretty simple. This is just showing you that how it worked with cytosine, which is a pyrimidine. Remember, so here it has the 1 position to the 1 position, and then this is a glycosidic bond, and this is what a glycosidic bond means. It's really easy. How are we going to link these two up? Okay.
Aye.
It's called a condensation reaction, also known as dehydration synthesis.
Aye.
Remember the ribose has the OH, the hydroxyl group right here, and the purine has the H down here. Okay, so what happens when these come together? What happens when you make H2O? What is that?
Water.
Water. That's why it's called a condensation. So this H here and this OH just go out. They condensate into water. So I'm just left with the nitrogen directly to the carbon here. See the nitrogen directly to the carbon right here? Remember, there's a carbon here. Just doesn't see it, but there's a carbon.
What do you mean nitrogen and carbon?
Okay, see this atom right here, the nitrogen on the 9th position?
Yeah.
Okay, when the H and the OH, the hydrogen and the hydroxyl, form water, this will link up to this carbon right here.
Okay.
Okay, and that'll form a glycosidic bond.
Aye.
And that's what we call a beta N glycosidic bond, or sometimes just an N glycosidic bond.
Aye.
Okay, so that's how these link up. Okay, so this is like I said, this is a condensation reaction.
Yeah.
So now we have an actual nucleoside like this. And
because the H2O like combined.
The bond forms when this water gets these can form to condensate to form water, and then nitrogen will bond here. So you have a sugar and you have the nitrogenous base.
Which is in DNA, right?
Which is in DNA. Don't worry about this slide yet. I'm gonna work on this slide. I don't think you can see it very well. It may not make much sense. Okay.
There's so many technical terms at once. It's just.
Yeah, I'm going over it fast, like I said, because this is my presentation. I'm just trying to do this for myself so I can kind of see where I'm at and maybe get some feedback on it.
Okay. H2O, nitrogen, glycosidic bonds.
Okay, so yeah, okay. Remember I said before there's a difference between the sugar between DNA and RNA. So people ask, what's the difference between DNA and RNA? Well, the difference is that DNA uses what's called deoxyribose, and RNA uses ribose. And the only difference is at the two position, ribose uses hydroxyl here, and DNA uses it uses H. So when you have the prefix deoxy, like deoxyribonucleic acid, deoxy means that a hydroxyl, an OH, has been replaced by an H. That's it. So on the two position, the OH has been replaced with an H. So this is what's going to be used in sugar, this in DNA, and this is what's going to be used in RNA, which we will get to in the second chapter, I think. Second presentation. So far we've constructed this much of it. We have the hydrogen bonds between the T and the A, and then we've added the sugar. So this right here would be the nucleoside, the nitrogenous base here, and the sugar here. So now we got to add. What's going on? Yep. Go ahead.
Purine?
Yeah, I can hear it. What?
The purine sugar, right?
No, the purine is the nitrogenous base.
Oh, really?
The sugar is a ribose. Remember, like in this case, deoxyribose, DNA, deoxyribonucleic acid. Yeah, this is actually a nucleotide because it has the phosphate. See the phosphate down here? We're going to add that right now. We're going to show what a nucleotide is. Because people say, what's the difference between a nucleotide and a nucleoside? Well, simple. A nucleoside is just the pentose sugar and the nitrogenous base. Okay? The nucleotide adds the phosphate. So these are actually nucleotides. Just different ways of looking at them. And let's go to the next one. So by adding, remember how we added the two the base and the sugar with the glycosidic bond, the beta 9. Okay, we're gonna do the similar thing with adding the phosphate group here, and that's called phosphorylation. Okay, phosphorylation means to add a phosphate group, right?
Okay.
And that's done by what's called a transferase, a phosphotransferase, or a kinase. Again, we already talked about these. Transferase will transfer something. In this case, we transfer phosphate.
Yeah.
A phosphotransferase is a specific kind of transferase, one that does a phosphate.
Yeah.
And then a kinase is actually a type of phosphotransferase, which is a movement. Yeah, movement. So these are all different classes, but they all transfer something. Okay. So what kind of bond do you think the phosphor is going to bond with the nucleoside with? Glycosidic, correct.
I didn't even say anything.
I know, but I knew you were going there because you're smart.
I mean, I know very little of what you mentioned. I mean, maybe I can say a term or so, but you know.
Well, let's show you why there's a glycosidic bond here. This is your ribose.
Yeah.
This is your phosphate group, right? This is another ribose with another phosphate group.
Wait, is the slide of what you're trying to point? Is the slide that is on related to what you're talking about?
Yeah, this is how we're going to be. Actually, no. You know what? It doesn't, because this is I don't add the phosphate here. Shit. Good point. All right, so I got to add a slide. to show how the phosphate group links. Because linkage is actually down here. No, I guess this works. I mean, this works, I guess. Let's add the phosphate. Okay, so this phosphate is going to link to here, okay?
Aye.
OH, OH. Okay, so if I take one hydrogen here and the OH out of here, what happens when I have H2O? Water.
Water.
Right? Okay, so here's my water going out. So I'm left with just this one oxygen atom right here.
Okay.
So this phosphorus will link with this carbon atom with an O linkage with the oxygen right here because water comes out of it. These are called ester bonds. So when Fiona talks about phosphodiester, what she's actually talking about is these bonds here, two ester bonds. So remember di meant two?
Yes.
So if somebody says phosphodiester, literally they mean what?
Two phosphorus. What? I don't know. Ester.
It's just two ester bonds, and ester bonds are here in a phosphorus. Okay, I'm not going to really explain what ester bonds really are because that would be another video. But these are just more the terminology. So phosphodiester is just these bonds here. Actually, without an oxygen is actually bonded with the double bond oxygen here. But I'm not going to get that complicated because I think I'll lose people. But if somebody wants to know what an ester bond is, I'll explain it to them. But anyways.
Aye.
So putting them all together, okay, to form a nucleotide, which is actually the monomer of DNA. You have your nitrogenous base, right? Adenine. You have your sugar, in this case ribose. This is not for DNA, this is for RNA, but it's in a picture I could find. But and then phosphate. So when you put all these together, you have adenosine monophosphate, also known as AMP. Excuse me.
All right.
Okay. Have you heard of adenosine? I'm sorry. What?
I feel so lost.
Well, let's take it back a step here, because I know I'm doing this really fast. One sec. Okay. Still see it? Can you still see the presentation?
No, I see you.
Maybe present again. Okay. Okay, so we're building up a nucleotide, and the way we build up a nucleotide is we have the nitrogenous base here.
Okay.
Adenine A, right? We have the sugar.
Okay.
Right, which is in this case ribose.
Okay.
And then we have the phosphate group right here. So going back to here, the phosphate, the sugar, and the base.
All right.
Pretty simple if you just look at it this way: phosphate, sugar, and base, right?
All right.
Which I may actually move this slide here because it should go with this slide. So this is
what?
This is what we call adenosine monophosphate. Have you ever heard people talk about ATP a lot on these videos?
Yeah, I've heard of it.
Yeah. ATP is the powerhouse of the cell. It's called adenosine triphosphate. So if it has triphosphates, how many phosphates do you think it has?
3.
3. So there'll be 2 more added on here.
Okay.
So this is the synthesis of a nucleotide. Remember, the OH will connect over here, and then the OH will connect over here. These form water. These form water. So 2 waters, and then you have a bond. And so these all link together. I'm not going to get into this stuff at the bottom right now.
Aye.
So we have how we name the nucleotides. This is where people get confused how we name the nucleotides. Again, we have the base, right? We have the sugar, and if it's OH, it's ribose. If it's H, it's dextroribose, right?
Yeah. Oh, shining. The shining. You got Jackson. Sorry, the guy who came in.
Down.
Is it down?
Down's corner.
Okay.
Okay, so.
Sorry.
All right, so this is our nucleoside, and then we have the nucleus.
It's not showing.
Damn it. Yeah, it should be.
It just okay. Wait, now I see you.
Now you see it?
No, I see you.
What the hell? Now?
Uh, no, I still see you.
What the hell?
So, so you,
No?
no.
Okay, this is pissing me off now.
So you need a phosphate, a nitrogenic, nitrogenous, and a sugar,
Um,
I guess.
is my mic bad? Can you hear me?
I can hear you.
Okay, somebody said that my mic is. Bob said my mic is still bad.
Uh,
In the side chat. I don't know. Okay, let me see why this is not working here. Stop presenting. Screen share. Okay, that should work there, right?
yeah.
All right. So if we're naming things by the nucleoside, then for each phosphate we add to it, we just add mono, di, and tri. Okay. So if we had like the nucleoside cytidine, we just add monophosphate, diphosphate, and triphosphate to it. If it's adenosine, we have adenosine monophosphate, adenosine triphosphate, or diphosphate, adenosine triphosphate. So that's all it is. It's the nucleoside and then how many phosphates it has.
Aye.
Now, if it's a DNA, like we're going to be working with, then all we do is add deoxo to it.
Aye.
And remember, deoxo was basically a hydroxyl being replaced to a hydrogen. So here's adenosine, right? Remember adenosine, the nitrogenous base.
Okay.
Deoxoadenosine, which is with the ribose, the deoxoribose.
Aye.
So when we start adding the phosphate groups, we just have deoxoadenosine, deoxoadenosine monophosphate, deoxoadenosine diphosphate, and deoxoadenosine triphosphate.
Okay.
So if it's RNA, we just have, remember, adenosine. If it's has a phosphate, it's going to be adenosine monophosphate and adenosine triphosphate. We just add this deoxo to it. That's how we build it up. Okay. So basically, it's going to look like this. You have your base.
Yeah.
Your nucleoside.
Okay.
You have nucleotide for RNA, nucleotide for DNA, and then your abbreviation. So if I have adenosine, my nucleotide is going to be adenosine monophosphate. For my nucleotide for my DNA is going to be deoxoadenosine monophosphate, and that's abbreviated AMP. If it's DNA, it's going to be abbreviated DAMP, right?
I guess.
Yeah, that little D there means DNA.
Okay.
So let's look at like cytosine. So cytosine, our nucleotide RNA is going to be cytidine. Remember, if it's a pyrimidine, it's going to end in DINE. So cytidine monophosphate, and then for nucleotide, it's going to be deoxocytosine monophosphate, which is called CMP or deoxocytosine monophosphate. And then the same thing for guanine and for thymine. Now, thymine is a little bit different. Thymine doesn't have a nucleotide for RNA. Why? Because thymine doesn't exist in RNA most of the time. It should. I think hypothetically it could, but
it don't exist in DNA.
Yeah, RNA replaces it with uracil.
Okay.
So it doesn't have one. So thymidine in nucleotide DNA is going to be deoxothymidine monophosphate, also known as DTMP. So this is the nomenclature that's used. So all you got to do to remember these things is break it down. If it's deoxo, you know you're dealing with DNA. Okay? And then if it's ending with sine or dine, it's going to be a nucleoside.
Aye.
So let's move on here. So basically, this is the overall picture. This is a little blurry. I might have to place this one. But it's basically, you know, your TA bonds with hydrogen bonds, C and G, your ribose group or deoxoribose group, and then your phosphate.
Aye.
And then I actually want to modify this picture because I want to show that these have a negative charge on the ends here.
Okay.
Can you see where my mouse is?
Can you move it around?
These are actually negatively charged, and that negatively charged is because there's a hydrogen atom. And
actually, try to put your mouse on like Pintos.
You mean the ribose?
Ribose. Oh, are you talking about the sugar?
No, no, I'm talking about the phosphate. There's actually there's a hydrogen donor that's actually a negative charge. That's why it's called an acid that I want to get into. So I need to explain that later. Again, I only did this overnight on my freaking tablet. I didn't even do this on a computer. And then I'll explain the 5' and the 3' end. 5', remember what we said about sugar? If it's 5', it's dealing with the sugar, right?
Yeah, I guess.
All right. So I'm gonna have to add a slide on that again. Hang on a second.
I don't feel that smart.
Well, remember and I'll make another slide to go in there. But remember the 5' end is this one, and the 3' end is this one. It's on the sugar. That's what they mean by 5' and 3'. And so when you look at the actual DNA, yeah, I'm kind of just doing this for myself, you know, because if I get it out there, I know what I need to fix on here. That's what all this is for. It shows the bases, right? How they're made, how they're put together in the double helix, and then this is actually shows what we're talking about. So this would be, you know, A. This would be T. So adenine, thymine, cytosine, guanine. This is your deoxyribose, sorry, and this is your phosphate. So this is called your sugar phosphate backbone. And the sugar phosphate backbone would consist of sugar, which is your deoxyribose, and your phosphate, which is your phosphate group that has your linkage to other nucleotides. So these are actually just, each one of these is a nucleotide. These are nucleotides are put together by phosphodiester bonds. So the sugar phosphate backbone is just built up by phosphodiester bonds. So that's kind of, you know, what I'm kind of going with this. Yeah, I mean, I know I went over it fast, but it was actually, like I said, for me, just actually to get it out there in my own head, what I need to actually look at. But I'm taking notes, by the way. So, do you kind of get into gist of what the building blocks of what DNA is going to be, though?
Yeah, sugar.
Yeah, well, it's going to be your nitrogenous base, your sugar, and your phosphate.
Yeah.
And then how they link together.
Yeah.
That's why I want to explain, you know, the glycosidic bonds and the phosphorylation.
Yeah.
And then when we get more into it, we're going to be explaining how the DNA actually gets read. And what happens, and basically the RNA polymerase will come in, they'll split the DNA, actually helicase splits it, but the DNA will split open, and then the polymerase will go by and it'll read this, and what it does is it codes it to what's called RNA, mRNA, messenger RNA.
Yeah, the uracil.
Yeah, what happens is if you have a thymine in the DNA, the RNA polymerase is going to be the complement of that, and the complement of that in the mRNA is going to be uracil. Correct.
Oh.
Yeah, see, you did pick up something.
Somewhat. I mean, it's like so much information. I'm bound to pick up like very little or something like that. Like.
Right, but okay. Let's see what you picked up just from that, though, because I went over this really fast. But if it ends in ASE, if it ends in A's, what?
An enzyme.
It's an enzyme, right?
Oh.
And enzymes are proteins. These proteins are enzymatic. So when you have like polymerase, it's actually a protein. When you have transferase, it's actually a protein.
Yeah.
Not all proteins are obviously enzymatic. Some of them are structural and other things like collagen is a protein. So.
Right.
But breaking it down, so if I say like transferase, you know it's an enzyme that's transferring something.
Yeah, I can write that down. Yeah.
Do you remember the difference between a deoxyribose and a ribose?
The type of sugar it uses.
The type of sugar it uses. So what is the sugar? What is the main difference as far as the structure between deoxyribose and ribose?
It has more different nitrogenous.
Well, remember that there's only on the 2' position. There's a difference in the atoms there. On a ribose, there's an H at the 2'. Now, at the deoxyribose, it's an OH.
Oh, it's the way the water is.
I'm sorry, I have it backwards. The ribose has the OH, and then the deoxyribose has the H. And what deoxo means literally is OH has been changed to H.
Okay.
Okay, so because OH has been changed to H, we call that deoxo. Okay.
Oh, okay.
I kind of want to get more into the nomenclature a little bit when I actually do this presentation with a bunch of people. The reason being is that especially like the nomenclature gets really confused to people when people say like, you know, what is adenosine 5 monophosphate? Well, we call it adenosine monophosphate, but it's actually adenosine 5 monophosphate because the phosphates attach to the 5' end of the ribose. So when you're looking at the structure and you see that 5', you have that CH2OH group coming out of it. That's where that phosphorus group is going to attach to. And that's why we call it. That's why it's actually called adenosine 5 monophosphate. But some of that stuff can get a little bit confusing, and it's not like, for example, hang on, let me see if I can bring it. I didn't add this, but maybe I'm going to add this to my presentation because I really want this to be demystified. Is the whole point. And some of the books I've read on biochemistry, they use a lot more technical jargon, but it's really not. Well, okay, let me say this. Biochemistry is the hardest freaking subject on the planet, without question.
Really?
Yeah, but I mean, seriously. I mean, biochemistry will make grown men cry. But some of the stuff what they use is not as bad as it looks initially. For like an entry level, so a first year would not be that complicated.
I feel like I should be able to spout everything you say, but it's just. Oh.
Let me see if I can. Let me see if I can find a name for this. I'm trying to look something up real quick. Dang it! I can't find what I'm looking for. You never find what you're looking for when you're actually looking for something, right?
Sometimes, maybe.
What else did you glean out of there, by the way?
Are you talking to me?
Yeah.
Okay. Nitrogenous, HOH, DNA, RNA, ribose, sugars, phosphates, triphosphates, diphosphates, monophosphates.
Oh, that's pretty good so far. Holy crap! You gleaned all that just out of that quick presentation?
Glyceride.
I didn't talk about glycerides.
Oh.
Yeah.
What you neurosides? I don't know.
Nucleoside.
Nucleoside.
Yeah, nucleotide. We do want to do. I talked to Drag, and I think we might do a video eventually on glycerides, triglycerides, starches. What a starch is, how glucose forms starch, how you have sugars made, monosaccharides, disaccharides. I think that'd be a good video. In fact, what's funny is I actually, it's cool. Is I actually started taking notes on sugar formations because I got a little ahead of myself, and I was like, okay, there's no possible way I can cover all this stuff, but I wanted to be able to explain to people what someone wanted to know how lactose was formed. So they messaged me the other day, so I was like, okay, well, let me take some notes on this so I can explain how lactose. But that's another video, but it looks like this.
Like milk, right?
Lactose in milk, yeah. But these are my notes on it.
Damn.
Yeah. Yeah. No, I got a shit ton of notes, man. I ain't kidding, man. I'm not even lying, man. I've been taking biochem notes like you wouldn't believe. So on this particular note, this is how the sugars would be. But this is actually what's called a beta 1,4 bond linkage. Okay. Remember, so if it's beta, we knew from this that it was the OH was on the same side as the main group, which is going to be here is going to be the CH2OH.
Okay.
So CH2OH, it's the same side on here. If it's opposite, remember it's alpha. So we know that this is on the same side. It has to do with antennomers, which is different ways of writing certain things. They're called stereoisomers, and I'm not going to get into that because it's complicated. But these two, this right here is a linkage between what's called beta galactose and beta glucose. So when you put these things together, they form a condensation reaction. And what is the product of a condensation reaction?
I know condensation deals with like perspiration and water.
Water, right. Water is the reaction, and you can't really see it, but I have an OH group, which is hydroxyl with a hydrogen. So this will actually produce water.
Okay.
Sucrose, which is table sugar, is actually just alpha glucose with a beta fructose, and that's all it is. It's called a dimer. So,
glucose, fructose, something.
Yeah, so if you have glucose and fructose, you have a sucrose, which is table sugar. It's a dimer of those two. So that's another video though. That's gonna be like part 6 of this series or something like that. So, hey Wolf.
Yeah.
We are boring the shit out of people with biochemistry.
Hello. What's up? Talking chemistry.
Yeah, I wanted to go over this so I could get in my mind what I need to do to fix this and change it and kind of get somebody like Course who's not really heavy into biology, to if I can explain to somebody on that level, no offense, Course, but I mean, if somebody's really not interested in this stuff, then maybe this video will do what I want to do, which is demystify some of these terms. People hear these, and the main reason I'm doing it is because we're kind of sick of people like no offense to true empiricism, but he uses these words to confuse people rather than educate, and that pisses me off. If you're going to use these words, you should be able to explain them and not try to confuse people by them. So I want to explain words like synthase, what it means, what is a transferase, what is a polymerase, what is a kinase, what is a phosphorotransferase. They're big sounding words, but what's that?
If you're looking for advocacy of scientific literacy, look no further. I would rather have them understand the processes of chemistry and the interactions that they play, than thinking that they were genetically engineered by ancient aliens.
Yeah, well, I'm kind of done with Arcane Logos, obviously, because he doesn't want to learn anything. I mean, he's just beyond the pale. So I mean, all he does is he's just a troll. It's just like, oh, you can't present evidence for evolution. I'm like, well, that's trivial, but I can present to you that 1 + 1 = 2, but if you don't know basic math, it's not going to do anybody any good now, is it? So, I mean, he's just that far removed from reality. He doesn't even understand basic science. But yeah, I've really been, like I said, I've been really heavily getting into this intro to biochemistry because I took chemistry, but I didn't, and I took biology. I got up to the Krebs cycle, so I actually had to learn the whole, glycogenic pathways and shit like that. But that was years ago, so I didn't have an interest in it as I do now. So maybe one of the videos where I'll do metabolic pathways like glycolysis, showing how it breaks down over like 10 steps to provide pyruvate and stuff like that, if anybody's interested. But I would have to relearn that shit myself. So.
Yeah, I do love my readings. Going all the way back to the first guy who ever came up with the concept of an atom, Democritus. It has definitely came a long way. Chemistry, ignored by I believe Aristotle, but sort of reinvigorated through alchemy, and then actually applying real general principles and how that stuff changes and shifts through laws of physics. I mean, it's really interesting.
Hey,
go ahead.
Sorry.
No, go ahead.
Okay, didn't Mendeleev come up with the periodic table?
Mendel? No, that was the gene. That was the gene.
Yeah, Mendel came up with ways of figuring out heredity.
No, I'm not talking about Mendel. I'm talking about the Russian who came up with the periodic table.
Oh, I don't remember who was that. I have to look that up. I don't remember.
I think it was Mendeleev.
I thought you were talking about Gregor Mendel.
Mendeleev. Yeah, Dmitry Mendeleev.
Yeah.
Yeah, Russian chemist. I actually remember that. I don't know why I forgot that. Unlike most people, if I don't know something, I just say, you know what, I don't fucking remember. I don't know, and I just go look it up because I don't know.
Mendeleev.
Yeah, it just sounds
weird.
I didn't want to start a couple people have suggested to me, they're like, maybe you should start at the periodic chart, because when we're talking about carbon and nitrogen and stuff like that, they may not know what that is, but I'm kind of assuming people have a basic level of this stuff, but maybe I'm kind of shouldn't. I don't know. What do you think?
I don't know. I feel like I'm below normal intelligence. Just,
Well, I wouldn't say that, but I mean, if we can explain it to you, then we should be able to explain it to anybody, because that's what the whole goal of this is, to explain to the average person that doesn't have an interest in this
so
what the stuff is.
yeah, so I'm like the very basic layman, I guess.
Okay, well, you know how to spell DNA. That's good, right?
Yeah.
So
Wait, are you talking about what it stands for, or just the acronym?
how to spell it?
Oh, like DEOXY.
No, DNA.
Oh, DNA. Yeah. Okay.
Simple.
Yeah.
But no, you remember? Do you remember what it stands for? DNA?
Oxyribonucleic acid.
Deoxyribonucleic acid.
Yeah, that's right.
And the reason why it was a deoxyribose and not a ribose was
The sugar.
the sugar is different, right?
Yeah. Like the H and OH thing.
The H and OH, right? The ribose has the H, and the deoxyribose has the H, and the ribose has the OH. So the OH is.
I'm not even sure what I'm talking about. I just memorized that.
It's fine, but you're right. I mean, that's what it is. So good job.
Okay.
Did you catch any of the presentation, Wolf? Wolf?
I'm sorry, I had a phone call. What was that?
Oh, I said, did you kind of catch the slides as I was going through them for your input?
I only recently came on the scene. I think you guys were mentioning. I didn't see the slides, but I think you're mentioning some terminology about like chemicals or something. Like I think it was related to heredity. I think.
No, I haven't gotten into heredity yet. I was just. I'm just going to present real quick and show you. I was just going over the slides to show how these linkages work, how we name these things, and basically what DNA is. I mean, how we look at these linkages, how we look at the nitrogenous bases, how we look at the sugar phosphate group, and take away the sting of some of the terminology. Because we look at this stuff and say, what is adenosine monophosphate? What is adenosine triphosphate? Well, it's just adenine, which is the nitrogenous base. We know that adenoside, which is going to be the nucleoside of it. Actually, is that right? No, hang on a second. I messed this up. I think I did. I guess I did this. I did it the other night. No, I got it right.
I think you did pretty good. I mean, like adenine, guanine, thymine, and cytosine. I mean, that does make up DNA.
And I think I made a mistake here. Now this should be adenosine. Adenosine. Did I? Adenosine. Yeah, so this should be adenosine. I don't know how I got adenosine there. It's a purine. See, but see how I figured that out though, that this should be adenosine, not adenoside, because it's a purine. Because a purine purine ends in ine, right? And a pyrimidine ends in deine. So this cannot be adenoside. It has to be adenosine, not adenoside. There's no such thing as adenidine. So just by knowing the prefixes, I know I made a mistake there. And this one needs to be cytidine, because cytidine is the nucleoside of cytosine, and that's a pyrimidine. So, anyways, see, it's kind of cool when you actually just know the prefixes and you go, oh crap, that's not quite right?
Yeah.
I can fix it on the fly. Not that anybody's ever interested in this, but. But like I said, the next one is going to be translation, and then there's going to be one on nuclear transport, which I have no idea about it. Nuclear transport is confusing. I don't know much about it. I understand the basics, how you have a protein that exists to transport across the membrane, a lipid bilayer lipid, but it's really hard. And then the next one would be translation, which I think I know pretty decently. I've had to explain codons and shit like that. That one I'm looking forward to, because I like the translation process. I think that's the kind of coolest. That's how the proteins are actually made there, of course. When you have, in the ribosome, you have amino acid chains being produced. It's called a polymeric chain, and that's coming out of the ribosome. That's going to form the protein. And that's a cool process, actually.
Amino acid change, polymer.
Amino acid chain is a polymer, yes.
Protein.
Protein is when it's completed, yeah.
I just I am probably forget that. But,
Like again, this whole series is when it's completed, it's really just to demystify these terms. That's all. So don't get too overloaded. I went through this really quick, and it was only for my own edification. Since I was making it on my tablet, literally I designed that on this, so it looks the same on the computer. It doesn't look the same on the computer, so I was able to see. Oh, well, I made the mistake there, and I could correct it. So.
I really feel like if I don't understand all of it, I feel not that smart, but you know.
Well, no, there's nothing to that. It's not as complicated as people make it out to be. The basics. Again, we're talking very basic.
Like diagrams.
Some of shit can get really complicated pretty quickly. Matter of fact, like I said, biochemistry is the hardest subject known to man. When you're dealing with metabolic pathways, it's freaking crazy, dude.
Yeah, like I guess it's tougher than physics.
Yeah. Oh yeah. Oh yeah. Yeah. Biochemistry is way harder than physics.
Oh really?
Yeah. Yeah. Have you ever seen a metabolic pathway chart?
Are you talking to me?
Yeah.
No.
Let me see if I can find it.
No. Does it have anything to do with like metabolism, like the way you digest and stuff?
So, yeah, well, yeah, it is metabolism. A metabolic pathway is metabolism. A metabolic pathway is how things are either broken down, which is one type of metabolic pathway, or brought up, built up from scratch. Anabolic versus catabolic. So catabolic is when they take something like what you eat and you break it down into its constituent parts. That's called catabolic. If you're building something up, it's called anabolic. And they have technical words, but I mean, when you know what they kind of do. When you hear like a catabolic pathway, you know you're dealing with somebody that's breaking shit down.
Did you say anabolic is when you throw up?
No. Break down catabolic and anabolic.
Okay, catabolic was like the constituency of what you eat, or I don't know.
Well, no. Think of catabolic, as catabolism. Think of a cannibalism. Well, when cannibalism, you're eating people. You're breaking down people. Yeah. So cannibalism is similar to catabolism, which is catabolic, meaning to break down tissue and shit like that. And for metabolic pathway. But I'm trying to find a metabolic pathway here for you.
And what's anabolic?
To build up.
Okay.
So I want to show you what I'm talking about here.
It's metabolic, catabolic, anabolic.
Yeah, we're not working on those terms yet, though. Those are for another video.
Oh, I'm sorry.
Here we go. Here's a good one. So I can make it bigger though. How can I make this shit bigger?
When you talk about metabolic, I think of like the hyperbolic time chamber in Dragon Ball Z.